Welcome, everyone. Welcome to the virtual roundtable, our consortium on translational research in the microbiome. My name is Joanne Geer, and I'm the Executive Director for the Westchester Biotech Project. Chris, would you just go on to the next slide? We are a nonprofit organization based in Westchester County, New York, and really are about developing collaboration across our local geographic community and the entire matrix of biomedical research and development, but of course national and international collaborations because it's a very small world. My partner, Michael Welling, is here today. Would you like to say a few words?
Absolutely. Good morning, everyone, and thank you for joining us and contributing to this topic, which, as you all know, is really becoming front and center throughout the entire research community. Just case in point, I had an introductory call this morning to a company based in Pittsburgh who is doing work on Barrett's esophagus and pre-cancer sort of indications, and microbiome came up as part of their research. So we know it's touching across the spectrum here, and we are excited to sort of help foster this collaboration. So thank you all very much.
Thanks, Michael. Chris, we could go to the next slide. I'm going to introduce Dr. Keith Bastian, who will tell you a little more about this consortium and his institute.
Thanks very much, Joanne, and welcome everyone. Some of what I'm about to say, probably those that have been on the previous call or involved in the organizing committee, this will be a repeat information. I'll just say a few words about ILSA and our interest in the microbiome space, and then what we're hoping to accomplish through this consortium. So I'm the CEO for the Institute for Life Science Entrepreneurship, and that organization is a nonprofit research institute and accelerator incubator that serves the New Jersey, New York metro area. We really focus our efforts in three areas. We work to accelerate innovative early stage technology, either from academia or already in startup companies, or from entrepreneurial scientists from industry. We have an incubator. We're located about 10 minutes from Newark Airport on the campus of Kean University. We have about 6 companies right now in our space, and we're working to expand our footprint to increase the space about 3- or 4-fold to 25,000 or 30,000 square feet of incubator space. And importantly, we have a research institute dedicated to translational science and in the advancement of research and translational science. We initiated the first Center for Translational Microbiology in a partnership with the American Type Culture Collection back in 2015, and we've had an excellent relationship with the ATCC, and that has brought the Institute into the throe of issues around standardization in microbiome research. We've also done work in antibacterial drug resistance and other aspects of industrial microbiology. So we actually have a research institute with scientists that are working in partnership with client companies, some in our incubator, others scattered around the world, working on their research programs, submitting grant applications, collaborating, and conducting our own research, mainly in the areas of applied microbiology. We're also just in the beginning stages of establishing a center for neuroscience, translational science. One interesting development that just recently happened, and there will be a press release on this in the very first part of February, is that we've joined the CARB-X initiative as an accelerator. CARB-X is a public-private organization, global organization that's working with BARDA, the Gates Foundation, Wellcome Trust, and other funding groups to provide non-dilutive research in support of antibacterial drug discovery and development They have over $500 million that they are investing in companies, in product developers to advance both traditional antimicrobial drug discovery and development, as well as non-traditional approaches. In addition, rapid diagnostics, vaccines, and other types of product development that are associated with this problem of antimicrobial drug resistance. So we're now working with a number of those CARB-X-funded companies and getting involved in a variety of very interesting research approaches to this problem. In fact, I just got back from a 2-day advisory board meeting reviewing grant applications up in Boston. And I should say that the world of microbiome and the AMR problem are definitely converging. And there were, in this particular round of reviews, there were a number of non-traditional approaches to antibacterial prevention and treatment. And a number of them were in the microbiome area, or phage-directed therapies to colonization targets for multi-drug resistance organisms in the GI tract of patients. And I should say that just my view is that there are a lot of very exciting things that are happening in this space, and the microbiome is playing, as you all are aware, is playing an increasingly more important role in how we think about infectious disease, just as one point. And why this consortium was set up, we've been organizing it and getting it off the ground for about the last year, is that there are a lot of issues around standardization, around big data analytical approaches and standardization of the informatic technologies, homogenizing and creating standard platforms for conducting research and analysis, dealing with the costs of doing research. But as the field is maturing and growing, and we've seen a remarkable progress over the last 4 or 5 years in this space in the translational science of the microbiome. So research directed towards product development that works through the microbiome has just grown tremendously, and the sophistication of the problems associated with this research have grown tremendously. So this is a sweet spot for our own research programs at ILSA, in our partnership with the American Type Culture Collection, and Dev Mittar is on the phone, and he'll be speaking from their perspective and some of the work that's being done there. Our particular interest is in the big gap between the in vitro or ex vivo experimentation that can occur around microbiome problems and what actually happens in the human gut. Our interests are predominantly focused on the gut, and we're interested in new model development in larger animal species that would be more predictive and validated models of the human gut microbiome, so that research could be done translationally somewhere in between doing a human clinical study and doing basic research. And we've been focused over the last year in developing a cost-effective, quote, cost-effective humanized gut mini pig model for studying microbiome interactions related to the human gut. What we're hoping to accomplish in the consortium is really to pull together a community of people who are involved, passionate, and at the cutting edge of microbiome research, and really begin to focus on some of these problems that we're all confronted with, but in a practical way. We're not looking to add to the wide number of meetings and events and conferences in microbiome, or to deal with company promoted events around their technology. We're really hoping to have a community of people that can work together in a very practical way, in a flexible way, in an ad hoc way through meetings like this, through face-to-face meetings, through webinars, to really identify and advance our understanding of these problems and hopefully create more networks amongst all of the groups that already exist in this space to try to make a difference in this one aspect of translational science. So, Joanne, I think I'll stop there. Next meeting, I'm not going to go through all this again. So, but I turn it back over to you.
Well, thank you. I think it's important to, I know they say you might be singing to the choir, but every once in a while the choir needs to sing together. So
right.
We may ask you to do it again. Things change every time. Dr. Vieira can't be with us today, but he has been instrumental in getting things off the ground. And he just arrived at Columbia a couple of months ago. So we'll be working together on profound interactions there as well. Chris, can you go to the next slide? We have a wonderful steering committee. I'll just leave this up while I tell you about what's going to happen next. We're going to hear from first Dr. Salomon Amar from New York Medical College, and then Dr. Tasha Santiago-Rodriguez from Diversigen. And each of them are going to speak for, up to about 10 minutes on the issues that we have raised. And after that, we'll go around and everybody on the call will ask you to introduce yourself and whatever you want to tell us about yourself and your thoughts about the content. You can certainly feel free to add questions to the chat box, but what we'll do is we'll have all the presentations first and then go around so everybody has an opportunity. The first two people that will be in that round will be Jessica Dunne from JDRF and Dev Mittar from ATCC. So I think we can go ahead and ask Dr. Amar to share your thoughts.
Thank you, Joanne, and welcome everybody. I'd like to extend thanks for inviting me to this consortium on translational research, as well as thanking Chris for doing all the legwork and putting all this together, as well as Keith. Wonderful presentation, and I'm looking forward to interacting with you guys at some point. I'm the Vice President of Biomedical Research. As you see, I have a dental degree, so and that's probably what earned me a spot over here, because not too many people work on this area of the microbiome, which is the oral microbiome. It exists, and it has some implication in regard to general health. Just as a housekeeping information, and Joanne did ask me to inform the group that we will be holding at New York Medical College, which is situated in the campus of Valhalla next to the hospital Westchester Medical Center. We will have a symposia on June 12, 2019 that will gather basic scientists, clinical scientists, and as well as the industry. And I scheduled the day specifically to capitalize on some of the presentation for sure, mostly on group discussions as well as general discussion, with the hope of generating at this point a white paper that could be given to NIH at some point, as well as published as the proceeding of the conferences. So I would welcome everybody to come and visit us and attend the conference, as well as collaborate with us. So New York Medical College is a 150-year-old institution with the medical school, now a dental school, a school of public health, and a nursing school with a strong affiliation with the Westchester Medical Center. A robust research program spanning from cardiovascular research to cancer research, stem cell research, as well as oncology and pediatric oncology. Essentially, we recently I moved to New York Medical College about 2.5 years ago, and I introduced originally an initiative that became progressively a center for oral microbiome and the microbiome at large. So it's in place right now. Just to mention that the campus has also an incubator on site, and we have now about 15, 16 tenants spanning different groups and different specialties. We're looking to, and we have the funds right now for the extension, which will extend to being able to accommodate probably 25 to 30 tenants. This incubator has been extremely prolific. Just recently, a company has received a Food and Drug Administration, an FDA approval, which is very exciting for that. They have their round for raising money. Each and every one has their aspect. So, as I said, the New York Medical College and especially the medical school has a robust research program. I wanted to focus it on the microbiome, and now it's in place. And that's what basically earned me this place in this consortium on translational research today. So several questions were raised to begin, hopefully, to engage into a deeper conversation at some point. And I'll focus on my area, especially the oral microbiome. The oral cavity is no matter how you clean the mouth, the minute you close your mouth after even a professional cleaning, you have a pellicle, a small precipitate that occurs on the teeth, and immediately you have a colonization of microorganisms, which constitute what we call the healthy microbiome. And then progressively, when left unattended, this microbiome that lodges itself between the gum and the tooth keeps on growing and becomes more and more pathogenic. And predominantly, I should say, as the pathogenicity increases over there, it becomes more gram-negative, filament-oriented or rod-shaped microorganisms. So we were interested into looking at what are the changes and how the microbiome over there can give us an insight into the pathogenicity of gum disease and caries diseases, especially as the mouth now being part and parcel of the GI system and cannot be separated from that. And we published a couple of papers recently, as well as early on, where we demonstrated that there is a change in the microbiome from health to disease. And we had a very interesting observation, and I'm going to summarize it because the papers have been published. There's multiple ways of being healthy in regard to the microbiome, but there are only few ways of being diseased in a sense, because you have drivers of microorganisms that cause a dysbiosis in the area and that drive the system constantly into a pathogenic mode. The questions that were raised by the organizers were DNA extraction, reproducibility, and the non-single omic methodology, which I'd like to spend some time. In regard to the microbiome in the mouth, or the oral microbiome, there's no so much specificity as compared to any other microbiome when it comes to the technology and the approaches for the analysis. So everybody started with the 16S RNA, and now moving to metagenomic and more in depth into the methodology for better read and deeper read into the sequences that we have. Clearly, DNA extraction is an issue because we might have, and we do, and everybody would agree with that, because the method that we have used, although they have been standardized by the manufacturers that we have that everybody is using, but we do have a human contamination from cells that are all around, especially epithelial cells, as well as some of the inflammatory cells that are all around, and that we take care of it when now we moved into metagenomic with softwares and platforms such as Migan or Mother that are allowing us to exclude some of those sequences. So carefully measuring the DNA output and standardizing the input DNA quantity across studies makes us achieve some level of standardization, although this is a work in progress, and I would be very welcoming into hearing what my collaborators on this panel, as well as the people attending, would have to add on this challenging situation. Reproducibility, randomness is an inherent characteristic of any open-ended approach, including the sequencing. So with the new technologies associated with increasing sequencing depth and better sequencing chemistry, definitely have contributed significantly to reduce sequence bias and improve the representation of the community that we are investigating. However, and I put it as an important however, the completeness of the database remain a challenge to adequately characterize and binding sequence data. The last question that was posed was the omic methodology can provide this new omic, no single, but the omic methodology can provide a complete picture of the biome under investigation. And with the integrated omic and targeted validation, especially when you obtain some of the results as the first approach, I think a more targeted approach using ELISA Western would and is still a mandatory validation for the microbiome aspect of it. But these targeted validation studies are helping us form a more cohesive picture of this community, and both must be carried out simultaneously to yield meaningful conclusion. I would leave it by saying that the metagenomic studies are highlighting that at this point, quantitative as well as qualitative microbial alteration occur in an increasing number of disease, and the oral disease are not excluded from that. Of course, they're included. Since the microbiome appears to be an actionable target for the development of both novel diagnostic tests, I should say, as well as possible targeted therapies. Just recently, as an aside, Akkermansia mucini was extracted from the GI microbiome, and this gram-negative bacteria, I'll give you a little bit of a story. It's a gram-negative bacteria. Now, when I trained in graduate school, a gram-negative bacteria is a bad news generally, but not anymore. This bacteria is a gram-negative, but that when supplemented in inflammatory reaction can counter the inflammatory reaction and even reduce bone loss associated with bacteria. So here you have possible targeted therapy. There's a growing interest that's been observed in this topic, but the enthusiasm is certainly tempered by the fact that most of the already published studies on the human gut microbiome report association with no clear data regarding functional effect. And that's where I'd like to stress that the endeavor of this group as well as other group should be put for a possible role on disease onset and development, which we have started in the oral disease, but which could be extrapolated to other phenomena. So the further development and optimization of these novel tools for data analysis, integration, correlation, and visualization will definitely allow us in the near future to fill this gap. In this way, it will be possible to understand functional, mutually beneficial relationship between humans and their gut microbiome, essentially now that aging becomes an important aspect that we have to focus on, and consequently the role of the microbiome in acquisition and maintenance of the healthy status. And I will probably leave it here.
Well, thank you very much. That's really a wonderful well, range of thoughts, and I'm going to be coming back to you saying, is this really what you said? Because you did a beautiful job of clarifying exactly why we're doing what we're doing. And I do want to say that for that June 12 conference, we've offered to participate, and Dr. Amar has graciously said that's okay. So as we have our conversation, we may come up with some interesting themes that can contribute to his agenda. Next, we're going to hear from Dr. Santiago-Rodriguez. And I will say there's some people on the call that haven't been properly acknowledged, and I thank you, Dr. Amar, for acknowledging Chris Kinzel. Chris is our program coordinator, and without him, there is no program. So I really appreciate his work. And Dr. Eric Vieira is with us today, but when we do our introductions all around, you'll get an opportunity, Dr. Vieira, to tell your story. Actually, maybe, are we transitioning? Actually, let me introduce you right now, Eric. Would you like to say a few words? I'm putting you on the spot.
Absolutely. Just to take a few seconds. I don't want to take too much more time, but happy that we're continuing on with this. I did make a transition from the CUNY system up to Columbia Engineering, and happy to participate in these forums going forward and figure out ways in which we can engage in the larger community. Columbia Engineering has 9 different departments, so we span many different areas. Engineering and medicine is an area of interest that we'll be exploring. Currently, we explore and will be continuing into the future. We don't have many researchers at the engineering schools looking particularly at the microbiome, but we have a lot of computer scientists and engineers who are very interested in understanding data and data science and applications. So I think that'll be very useful for microbiome research going forward in some of the work that we hope to bring forward in this forum. So I'll end there because I really want to get back to the main discussions of our conversation today.
Thanks, Eric. Tasha, you may have something on your screen asking you to show your screen. You've been made, yeah. Or did we decide that Chris was going to run your slides?
I think I was going to.
Here we go.
Oh, okay, here it is. Can you see them? Can you see the slides?
It's, I have a funny screen. Does anybody see the slides? No. You know what, Chris, why don't you go ahead and run the slides? Because we know it works from your side. And Tasha, you can just tell him when to change and he'll go ahead for you.
That sounds good.
Great. And Mr. Monza, I see that you have a question there. We'll get to that. You know, we'll address the questions after we have this next talk. Go ahead.
Okay. I wanted to give you a thank you for the opportunity to speak here today. Next slide, please. Before I talk about some of the topics about microbiome research and some of the challenges that we have encountered, I wanted to talk very briefly about Diversigen. Very quickly, Diversigen is a microbiome service company based in the Texas Medical Center. It is building on research conducted at the Baylor College of Medicine in the Center for Metagenomics and Microbiome Research, and has been operating since 2014. Next slide. So as all of us know, microbiome research has expanded exponentially. That's one of the reasons why we're here. And as the field grows, we have realized that there are challenges or biases that can be introduced at different stages of microbiome research, from sample collection and storage to nucleic acid extraction, not yet, nucleic acid extraction, library prep and sequencing, and data analysis. Next slide. I wanted to talk about some of the challenges within sample collection and storage, which is probably one of the first steps after designing the study. There are different practices for sample collection And some of these practices will depend on the type of sample. There could be variations in some of these practices. And what happens is that these variations can result in variations in the results. I learned very recently that the microenvironment within a sample can also affect microbiome results. There was a study that found that if you sample a stool sample inside or outside the sample, it can affect the detection of certain bacteria. For this reason, it has been suggested to homogenize the stool sample before DNA extraction and sequencing. Once a sample is collected, another factor that can challenge microbiome studies is the storage temperature. It is widely suggested to immediately freeze a sample at -80 degrees for long-term preservation. And what happens is that when you store the sample at higher temperatures, such as -20, 4 degrees, and even room temp, there's the risk of variation in some of these results, including variations in the alpha diversity and some of the taxonomic relative proportions. Other studies may require a preservation buffer if there is no immediate access to a -80 freezer. There are different types of preservation buffers, such as RNA later, OmniGene gut. These are just examples. And unfortunately, each of these buffers can preserve a microbiome sample differently. There was a study that showed that OmniGene gut can preserve a sample similarly to a -80 freezer. And then another study found that RNA later can actually decrease the abundance of certain bacteria. Another factor that can influence this particular step of microbiome research is the number of freeze-thaw cycles, which can affect the abundance of certain bacteria. So it's not really recommended to use a sample after multiple freeze-thaw cycles. Next slide. Also, the nucleic acid extraction is, of course, something that can add bias to microbiome research and has been addressed in multiple studies. The method, of course, will depend on the sample type of interest. There are traditional methods such as the phenylchloroform method, which can be cheaper, but then it takes more time, or there are commercially available kits. Nevertheless, the microbiome community understands that one of the major things to consider when you process a microbiome sample is the cell wall composition of the microorganisms, mainly gram-positives and gram-negatives. For example, a stool sample has a mixture of both gram-positives and gram-negatives, and since gram-positives are hard to lyse, it will require additional lysis steps such as mechanical shearing, like bead beating, or enzymatic lysis, such as the addition of a lysozyme or protein escape. And what happens when some of these steps are not applied, the number of species identified tends to be much lower. Another thing that can affect microbiome studies within this area is the DNA concentration that you use for library prep. There was a study that showed that the number of OTUs or species tends to decrease with a decrease in DNA concentration. Another aspect that can affect microbiome research within this area is the host DNA depletion, which is very important, especially when handling samples that are highly impacted by host DNA contamination, such as skin samples, saliva samples, blood samples. And the problem is that when we don't add that particular step, the vast majority of the sequences can correspond to the host genome. Next slide. Library prep protocols and sequencing platforms can also add biases to microbiome studies. For example, although there are standardized protocols for different regions within the 16S ribosomal RNA gene, like for the V1, V2, V4, V3, V4 regions, sequencing different variable regions can also add biases to microbiome research because usually you tend to get different taxonomic proportions. The library prep protocol can also affect microbiome studies. There was a study that tested different library prep protocols, including the Illumina TrueSeq, Nextera XT, and 2-kappa hyperprep protocols. Some of these include a PCR step, some of them don't. And what this particular study suggested is to use a PCR-free method because it may provide more accurate results. Also, the technique used to get the fragments either if it's enzymatic or mechanical shearing, for example, can also affect the output in microbiome studies. In terms of the sequencing technology, we know that Illumina sequencers have become the preferred choice, but a lot of studies in the past, and still some of them, have been performed using 454 sequencing and the PGM from Ion Torrent. And there is really an increased interest in combining data sets to find patterns across different data sets. And there are researchers that suggest that this can be done if the same PCR primers and bioinformatic workflows have been applied. And also people suggest that the variations between the methods must be quantified and they need to be accounted for when interpreting the results. And another thing that can affect microbiome research within this particular area is the sequencing depth, which I'm going to be talking a little bit more about some of the studies we have done here at Diversigen. But it has been considered in the past mostly for 16S studies. And some of these studies have shown that with an increase in sampling depth, there is a decrease in variation in the data. Next slide. And finally, for this part, the data analysis, as all of us know, is a big influencer in microbiome studies. For example, the availability of groups to compare your data with, the availability of metadata can affect microbiome studies. Also, the taxonomic classifier is a huge influence in microbiome research. The database use or the lack of a database, of course, can impact microbiome research. Next slide. And as I mentioned, I wanted to expand a little bit on the topic of sequencing depth. This is something we have been doing here in Diversigen. As you know, it is very hard, even with our current sequencing technologies, to fully capture all the DNA sequences from every microorganism in a sample, particularly if it's a very complex sample, such as soil or a stool sample. And even though the cost of shotgun metagenomic sequencing has gone down, it's still relatively high compared to 16S. So there is still a limitation in the amount of data that can be acquired. And this in turn may impact the biological and ecological interpretation of microbiome results. So it's very important to estimate the required sequencing depth that is needed to have meaningful results in order to characterize a microbiome sample in terms of the taxonomic and functional composition. Next slide. So this is some of our data here at Diversigen to determine, we wanted to determine the potential effect of different sequencing depths in microbiome studies. So for this, we collected 10 human stool samples. We sequenced them to a depth of about 5 gigs of data, and we in silico subsampled the reads to mimic a different sequencing depth. As you can see here with the different colors, each of them represent different sequencing depths, and the 16S was also performed just to compare. We performed alpha diversity analysis that included the observed genera and the Shannon diversity index just to see the evenness. We also added to ATCC mock communities MSA 1001, which is composed of 10 strains in even concentrations, and MSA 2002, which has 20 strains in even concentrations. And what we found is that the sequencing depth doesn't seem to affect the mock communities, possibly because there is a simple community, but it tends to affect the alpha diversity of the stool samples, possibly because it's a more complex sample. When compared to 16S data, as you can see here, shotgun metagenomic data have a lower number of genera, and this is possibly because of the number of genomes available in databases compared to the number of 16S sequences available in the databases. So that's probably one of the reasons why there's a huge difference there. In terms of the evenness or the Shannon diversity index, the different sequencing depths don't seem to affect the evenness of the mock communities or the stool samples. In terms of the beta diversity in panel A, we see that as expected, here you see on the right side, the stool samples, the pink represents the 16S sequencing of the stool samples, and the remaining represents the metagenomic sequencing, there's a separation of the data based on the type of sequencing, which is expected. Also, you can see the mock communities clustering separately from the stool samples, which is expected. But when we look at the data only at the stool sample beta diversity, as shown here in panel B, we see that there are significant differences based on sequencing depth. Next slide. In terms of the taxonomy, I'm showing you the data on the left at the phylum level and at the genus level on the right. At the phylum level, it seems like the sequencing depth doesn't affect the relative abundance of these 5 different phyla represented in the graph for the mock communities or the stool samples. So it seems to remain pretty constant throughout the different sequencing depths. But at the genus level, different sequencing depths seem to affect the identification of rare genera or those present in low relative abundances, as shown here in panel C. Each of these colors represent a different sequencing depth, and what happens is that some of them are detected at higher sequencing depths, but as you go lower, they are no longer detected. Unlike some of those genera that are present in high or medium relative abundances, you can really detect them at the different sequencing depths. And as probably expected here in panel D, you can see that there are some genera that are only detected when using 16S sequencing, possibly because of the differences in database composition. Next slide. We also saw a similar trend with the pathways. These are functional pathways. I'm not showing you the names of the pathways, but it's just to have the idea that with lower sequencing depth, there seems to be an effect on the identification of some of these, as you can see here in panel C, but it doesn't really happen with those that are identified at higher and medium relative abundances. So the take-home message of both the taxonomy and the functional data is that different sequencing depths may affect the reproducibility and interpretation of the data concerning these genera and pathways that are present only in low relative abundances. Next slide. And I wanted to finish, and I'm sure, we're going to be expanding on this a lot. The take-home message is that we all know that microbiome research is still expanding and it's evolving, and as the sequencing, the analysis, and databases capabilities expand, the studies will probably continue to differ across different labs. Nevertheless, we need to standardize somehow microbiome research with what we have right now. And this can be done, for example, by adding controls such as mock communities and spiking controls, which I know ATCC is working on. Some people prefer well-characterized stool samples. There's also the need to carefully address and measure some of these biases that can be introduced at different stages of microbiome research. Like, for example, some of the data I've shown you today with the sequencing depth, there's the need to probably address most of these biases that can be introduced at different stages. And with this information and the studies, we need to come to a consensus and then determine which practices are the best for both 16S and WGS or metagenomic sequencing. Next slide. I just wanted to thank you again for this opportunity, and I wanted to thank the Diversigen Science and Data Analysis team. And also at the end, the next slide, there are some references if any of you are interested in some of these studies I've talked about.
Thank you very much. And we can make these slides available as well as the recording on our archive channel. Excuse me. What a great beginning. This is terrific. Thank you so much. We will hear briefs from 2 additional people, and then we'll have a moment to be able to go around and let everyone introduce themselves briefly and talk about what this discussion means to you and anything else. If you plan, you're gonna have about a 2-minute window to tell your story, and hopefully some themes will come up that we want to dig into a little more deeply. Next, we're going to hear from Jessica. You've got 5 minutes, so you go ahead.
Thanks, Joanne.
Jessica Dunne, Dr. Jessica Dunne, Director for Discovery Research at JDRF. Welcome, Jessica.
Thanks, Joanne, and thanks, Chris, and thanks everybody, all the organizers as well for putting this together. This has been great so far. I will try to be brief in my comments, and I really think, there have been some great themes that have come across so far from Dr. Amar and Dr. Santiago-Rodriguez, in terms of how we do our analysis, and what the right ways to do it are, whether or not we can use the microbiome for diagnostics or for therapeutics, and really intrigued by some of the themes that Dr. Santiago-Rodriguez just finished on with, standardization of microbiome research to be sure that what we're getting, what we're publishing as our results are really meaningful, because I think, all of that will affect the themes that Dr. Amar brought up with respect to moving forward with the microbiome as either diagnostics or as therapeutics. And so JDRF has, we are a nonprofit, the world's largest private nonprofit that does type 1 diabetes research, and sort of coming on the heels of other autoimmune diseases. Back in 2011, we launched our Microbiome Consortium to really understand, if the microbiome plays a role in the development of type 1 diabetes. And long story short, I think we are convinced that it does. And about 3 or 4 years ago, we also were feeling the pinch in terms of how do we, are we really doing the right things to lead towards the diagnostic and the therapeutic areas? And one of the things that we noticed within our consortium, we had actually made data deposition a requirement for public data deposition of a requirement for all of our awardees, all of our funded investigators. And we really felt that the existing infrastructure was not fulfilling our needs. So, your 16S sequencing was going here, your whole genome sequencing was going there. If you had any clinical metadata, maybe or maybe wasn't associated. And if you were looking at any immune cells or any other types of analyses, it was impossible really as a third-party observer to pull the studies together and make sense of them. And so we started 3 years ago our microbiome, what we're calling the Microbiome Initiative Bioinformatics Center. And it's led by Curtis Huttenhauer at Harvard, who is also part of the HMP and HMP2 programs. And really what it is first and foremost, it's infrastructure that allows for data deposition of multiple data sources, both including mouse, human, other rodent models, and also really provides a resource for our investigators in terms of computational resources. So they have the ability to utilize either the analytic team at Harvard or utilize the tools that exist on the Bioinformatics Center for their own analyses. And one of the other things that it enables, because of our data sharing rules, we can cross-compare different studies. Again, we provide the computing infrastructure. There's also some support in terms of harmonizing data storage, annotation, and distribution. And so the portal is secure and it's authenticated. And then there are ways to disseminate based on some, so basically if you write a paper and you have been depositing your data according to JDRF's rules, by the time it gets to public data release, the Microbiome Informatics Core will do that public dissemination for you. One of the other things that we are doing, and this is relevant to what Dr. Santiago-Rodriguez was talking about, was developing some SOPs for our community that also are available on the website. So, I think actually my 5 minutes is just about up, but I think what one of the things that we are really keen on is making sure that this theme of standardization and also that the right tools in terms of the analysis are happening. I think one of the concerns we've heard is that sometimes folks aren't using the right in analytical tools to put forth the analyses, and I don't know if there's any such thing as a perfect analytical tool. And in fact, if many of you saw the Nature papers that were published by the Teddy group back in the fall, one of the reasons it took a long time was because the two groups looking at the data were analyzing the data in different ways and coming up with slightly different themes for the data. And so what they did is they spent a lot of time coordinating in terms of how they were doing the analysis, how they were cleaning their data. Some of the themes, again, that Dr. Santiago-Rodriguez was talking about in terms of host contamination. So all of that was, brought to bear. So there is a public website, and some of the protocols for what the teams are doing are made public. So if you Google JDRF MIBC, and I think I sent the URL to Chris, so we're happy to send that out as well with information. Take a look on that, and feel free to utilize any of the public resources that we have available as well. So thanks, and pass it back to Joanne.
Thank you very much. It's really great to see where you're at, and I'm familiar with a couple of people on the call, and I imagine your wheels are buzzing at the idea of really putting in the energy to create that infrastructure for computation from the beginning. I'm sure it's making a huge difference. So that's terrific. And yes, we're starting a resource page, and I will mention those of you on the call who have something that, like your own website or a paper or something you think would be interesting, we'll be very happy to post it. I want to introduce Dr. Dev Mittar from ATCC.
Hi, everybody. I really enjoyed the discussion. That's very thought-provoking, and it's very satisfying to know that the value of standardization. Tasha and I used to work together, and we have developed some standards together as well. And I'm glad to see her work on standardization using ATCC standards. So the field is really moving from academia to translational microbiome now. Initially, we came up with a set of 6 standards where we mixed a very diverse set of bacteria to create mock communities, and I'm really happy to see a lot of academic and industrial folks are using those standards and comparing data against, and it is really helping. We got a lot of feedback, like the work that Dr. Amar mentioned, the very applied work on oral, skin, gut, and vaginal microbiomes. There is no standard which is perfect, which can really mimic a actual microbiome specimen. So, but given the feedback that we got for those translational studies, so we put together some specific standards which are geared towards oral microbiome, gut, skin, and vaginal microbiomes. For example, we have created a standard for which contains organisms that are commensals in oral community as well as involved in disease state. I'll tell you briefly, for example, like actinomyces, which are present in dental plaques. We added gram-positive versus gram-negative. For example, Fusobacterium is one of the periodontal pathogen. And then there are common commensals such as Haemophilus, Prevotella, the Streptococcus mitis, which is present in dental caries. So the feedback that we got from all those people who are working on clinical trials, translational studies, that they have need for a standard or a control which mimics specific microbiome specimen. So we are happy to tell you that those standards are now available from ATCC collection. We have done a lot of beta testing with people like Tasha, Santiago, and we'll be happy to help any other who has a need for some specific mock community. So right now, in addition to 6 generic standards, we have oral, skin, gut, and vaginal microbiomes. Plus, we are also working on spike-in standards and a fungal mock community and a viral mock community as well. Maybe in next talk or something, I can present some of the spike-in standards work that we have accomplished so far.